This lecture is on Chapter 1, the study of body function. We'll start with Chapter 1.1, the Introduction to Physiology. Physiology is the study of biological function, which is how the body works. Physiology is concerned with the normal function of cells to the organism as a whole. Physiology emphasizes mechanisms, how it works. Physiology explains using cause and effect sequences, and physiology is derived from scientific experiments. Pathophysiology is concerned with how disease or injury affects physiological processes. Pathophysiology aids in understanding of normal processes. Comparative physiology studies the differences and similarities in the functions of invertebrates and vertebrates. Comparative physiology has aided in the development of pharmaceutical drugs through animal trials. The scientific method is used to make observations, form a testable hypothesis, design and conduct experiments or make more observations, and analyze the data. The results must be replicated many times before a conclusion is accepted. Several verified hypotheses may become a general theory based on reproducible data. In the scientific method, there are measurements, controls, and statistics. Good physiological research requires quantifiable measurements, an experimental group and a control group, a reduction of bias, statistical analysis, and review and publication by a peer-reviewed journal. When developing pharmaceuticals, basic research is conducted for years before a drug is ever given to a person. Research begins by studying the effects of a chemical on cells in vitro, which means in a culture dish. Next, the studies are done in animals, usually rats and mice, to see if the same effects occur in vivo in a living creature, and if there are any toxic side effects. For these trials in developing pharmaceuticals, many rats and mice are genetically modified to be susceptible to particular diseases. Animal trials may take several years. There are several phases of clinical trials. In Phase 1, the clinical trials test the drug on healthy human volunteers to test for side effects, rates of passage, dosage, etc. Next, the drug goes into Phase 2 clinical trials to test its effectiveness on people with the particular disease. In Phase 3 clinical trials, these are conducted on a large number of people to include both sexes, many age groups and ethnicities, and people with more than one health condition. From here, the FDA can approve the drug for sale. Phase 4 trials test other applications for the drug. Chapter 1, Section 2 describes homeostasis and feedback control. In the brief history of physiology section, the book discusses several great thinkers: Aristotle, William Harvey, Claude Bernard, and Walter Cannon. Through their work, the term homeostasis was coined, and homeostasis describes the internal consistency of the body. Thus, homeostasis is the constancy of the internal environment. And the main purpose of our physiological mechanisms is to maintain homeostasis. Deviation from homeostasis indicates disease, and homeostasis is accomplished most often by negative feedback loops. In the negative feedback loop pathway, the receptors act as sensors in the body, and they detect a change and send information to the integrating center, which assesses the change around a set point. The integrating center then sends instructions to an effector, such as the muscles or the glands, which can make the appropriate adjustments to counter the change from the set point. Mechanisms of negative feedback loops move in the opposite direction from the change. Negative feedback loops make the change from the set point smaller, they reverse the change in the set point, and this is a continuous process, always making fine adjustments to stay in homeostasis. In figure 1.1, shown on the left-hand side of the page, the illustration states that there is a rise in some factor of the internal environment indicated by the arrow moving up and X. So whichever variable that could be, temperature, blood pressure, blood pH, sugar levels, cholesterol, insulin, you name it. Things that are going to change in your internal environment as the external environment puts pressure on them. So in this case, we see a rise in the factor, and it is detected by the sensor here in purple. This information is relayed to an integrating center, shown here in yellow, which causes an effector, the blue, to produce a change in the opposite direction. Thus, our factor decreases back to set point. The initial deviation is thus reversed, completing a negative feedback loop, shown by the dashed arrow and the negative sign. That's our negative feedback loop. The numbers indicate the sequence of changes. In the next figure, figure 1.2, we see a fall in some factor, indicated by the arrow going down in X, of the internal environment detected by a sensor. Taken to the integrating center for processing, a decision is made which goes to the effector organ, which carries about the process of reversing the change in the stimulus and bringing factor X back up to normal levels, thus completing the negative feedback loop. The take-home message here is that the negative feedback reverses the change in the stimulus and takes us back to our normal set point. And the change in the stimulus can either cause a rise in the factor or a drop in the factor. Either time, the integrating center has to decide whether we're rising or falling and what we need to do about it in order to reverse the change of that rise or fall and take us back to our set point. This is the basis for all the blood tests that you go and get when you go to the doctor and they're testing for your sugar and A1C levels and your cholesterol and triglycerides and vitamin D and iron and hematocrit. All of that is going to be maintained through these negative feedback loops. And if they are imbalanced, as stated earlier, that can lead to disease. Here's another example of a negative feedback loop that is usually given in an anatomy and or physiology course. Body temperature. So sensors in the brain will detect the deviation from 37 degrees centigrade, which is our normal body temperature. Another part of the brain assesses this as actionable, and the effectors, which are sweat glands in this case, are stimulated to cool the body. Once the body is cool, sensors alert the integrating center and sweat glands are inhibited. The end result regulates the entire process. Production of the end product shuts off or down-regulates the process. This is why it's called a negative feedback loop. By reversing the change in that stimulus, the end product returns us to our set point and the system is shut down because we no longer need it. Until next time. There are antagonistic effectors that often maintain homeostasis, and these are opposing effectors that move conditions in opposite directions. and this maintains conditions within a certain normal range or a dynamic consistency or constancy. For instance, when you are hot, you sweat. When you are cold, you shiver. These are antagonistic reactions. Other examples are blood glucose levels. When your blood glucose levels rise, your pancreas secretes insulin. When your blood glucose levels fall, your pancreas secretes glucagon. We have blood calcium levels, and the hormones parathyroid hormone and calcitonin antagonize each other. With blood pH, we have carbonic acid and bicarbonate ion. In figure 1.3, we see that negative feedback loops maintain a state of dynamic constancy within the internal environment. The completion of the negative feedback loop is indicated by the negative signs. With the body temperature in figure 1.4 featured on the bottom of this slide, it says how body temperature is maintained within the normal range. The body temperature normally has a set point of 37 degrees Celsius, and this is maintained in part by the two antagonistic mechanisms, shivering and sweating. Shivering is induced when the body temperature falls too low, and it gradually subsides as the temperature rises. Sweating occurs when the body temperature is too high, and it diminishes as the body temperature falls. Most aspects of the internal environment are regulated by the antagonistic effects or actions of different effector mechanisms. Quantitative measurements are important in negative feedback loops. In order to study physiological mechanisms, scientists must measure specific values and mathematically determine such statistics as their normal range, their averages, and their deviations from the average, which can represent the set point. A knowledge of normal ranges aids in diagnosing diseases and in assessing the effects of drugs and other treatments in experiments. Table 1.2 includes approximate normal ranges for measurements of some fasting blood values. In this table, you see it's in your book, so you can refer to that. It's blue in your book and peach colored here. But we've got arterial pH, our bicarbonate ions, sodium ions, calcium, oxygen content, urea, amino acid composition, proteins, total lipids, and glucose. Positive feedback is the other type of feedback loop. It is less common by far than negative feedback. Negative feedback makes up about 99% of homeostatic feedback loops. So about 1% of homeostatic feedback loop include positive feedback. And in positive feedback, the end product in a process stimulates that process further. Thus, the action amplifies the changes that stimulated the effectors. Positive feedback could not work alone, but it does contribute to many negative feedback loops. For example, if a blood vessel is damaged, a process has begun to form a clot. Once the damage is fixed, clotting ends, which is negative feedback. However, the process of forming the clot involves positive feedback. The strength of uterine contractions during childbirth is also regulated by a positive feedback loop. The regulation of homeostatic processes within organs can occur in two ways: intrinsic and extrinsic regulation. In intrinsic regulation, cells within the organ sense a change and signal to neighboring cells to respond appropriately. In extrinsic regulation, the brain or other organs regulate an organ using the endocrine or nervous system. The endocrine system releases hormones into the blood and transports them to the target organ. That's what the blood does, transports throughout the body. The nervous system will innervate organs with nerve fibers. Illustration 1.6 shows the negative feedback control of blood pressure. Blood pressure influences the activity of sensory neurons from the blood pressure receptors, which are your sensors. So here we see the stimulus, number 1, as the blood pressure falls, and the sensors in number 2, the blood pressure receptors respond. A rise in pressure increases the firing rate, and a fall in pressure decreases the firing rate of nerve impulses. In this case, the blood pressure has fallen, and so it has decreased the nerve impulses. When a person stands up from a lying down position, the blood pressure momentarily falls. The resulting decreased firing rate of nerve impulses in sensory neurons affects the medulla oblongata of the brain, shown in the integrating center. This causes the motor nerves to the heart, which will be the effector, to increase the heart rate, helping to raise the blood pressure. So as the heart rate increases, the blood pressure increases. My apologies. Feedback control of hormone secretions. Hormones are secreted in response to specific stimuli. For example, an increase in blood sugar results in the release of insulin, which removes sugar from the blood. Secretion can be inhibited by its own effects. For example, decreased blood sugar inhibits the release of insulin. Negative feedback inhibition is a closed-loop control system and usually involves an antagonist to make sure homeostasis is maintained within normal levels. For example, when blood sugar is low, the hormone glucagon is secreted, which results in a rise in blood sugar. So this is antagonistic to insulin. In figure 1.7, it shows negative feedback control of blood glucose. In figure A, the rise in blood glucose that occurs often eating after carbohydrates is corrected by the action of insulin, which is secreted in increasing amounts at that time. In figure B on the right, during fasting, when blood glucose falls, insulin secretion is inhibited and the secretion of an antagonistic hormone, glucose, is increased. This stimulates the liver to secrete glucose into the blood, helping to prevent blood glucose from continuing to fall. In this way, blood glucose concentrations are maintained within a homeostatic range following eating and during fasting. Chapter 1, Section 3 discusses the primary tissues. In the levels of organization of the human body, the cell is the basic unit of structure and function. Cells are groups, or tissues are groups of cells that are similar in structure and function. Organs are a group of 2 or more tissues, and an organ system is a group of organs that work together to perform related functions. And the organism includes all the organ systems working together in coordination. Our organs are composed of 4 major categories of tissues: muscle tissue, nervous tissue, epithelial tissue, and connective tissue. Each tissue has particular structures and functions that dictate the physiology of the organ. Muscle tissues are specialized for contraction, and the 3 types are skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is a voluntary muscle, which is muscle you can consciously control. We often call this somatic. Skeletal muscle tissue is mostly associated with the bones that are pulled to produce movements. A few skeletal muscles do not cause skeletal movements, but are under conscious control anyway, such as the tongue, the esophagus, the sphincters, and the diaphragm. Skeletal muscle tissue has visible striations from sarcomeres, which is the layout of the contractile proteins. During embryonic development, the union of separate cells called myoblasts form myofibers. Myofibers are the muscle cell, and they are considered a syncytium, meaning that they are a multinucleated cell. Pardon me again. The muscle cells can produce a graded response that increases the strength of contraction as needed. This illustration shows skeletal muscle tissue, and you can see the muscle fibers are multinucleated and striated, and they are going to be under voluntary conscious control. They're what we call somatic. Cardiac muscle tissue is found only in the heart, and the fibers are short, branched, and interconnected both physically and electrically. Cardiac muscle tissue is striated, but is very different in the structure and action from skeletal muscle. Cardiac muscle tissue is involuntary. You cannot consciously control it. It is specialized to allow for the passage of sodium between the cells. That helps the cells to beat in unison. And so these are cell connections called intercalated discs. They're a special type of gap junction. Cardiac muscle tissue cannot produce a graded contraction. This illustration is a photomicrograph of cardiac muscle, and you can see that the striated appearance and the dark staining intercalated discs are evident. This is figure 1.9. Smooth muscle is found in the walls of digestive, urinary, and reproductive organs, the blood vessels, and the bronchioles of the lungs. These are all hollow organs. Smooth muscle tissue is involuntary, like cardiac muscle. However, it is not striated, which is where the name smooth muscle comes from. Smooth muscle tissue is generally found in layers that run in different directions. There will be longitudinal muscles, circular muscles, and even diagonal muscles. Peristalsis is the coordinated wave-like contraction of smooth muscle layers to move substances through the organs. That's a typo there. It's not more, it's move substances. This illustration, figure 1.10, shows smooth muscle tissue. And you can see that it's a single nuclei in each of the cells, no striations, and the cells are going to be small and pinched on both ends. The next tissue is nerve tissue. Nerve tissue is found in the brain, the spinal cord, and the nerves. It's composed of neurons and neuroglia. Neurons conduct impulses and have three parts. The dendrites are short, highly branched cytoplasmic extensions that receive signals. The axon is a long single extension that sends the signal, and the cell body is the metabolic center containing the nucleus. Neuroglia are supporting cells that do not conduct a nerve impulse, but are essential for nerve function. In fact, they outnumber the neurons. This illustration shows a large motor neuron, and you can see the numerous smaller neuroglial cells surrounding the motor neuron. Neuroglia means neural glue, in essence. Neuroglia. Sorry, I'm moving around so much. Sit still, Cooper. All right, epithelial tissue. Epithelial tissue forms the membranes that cover body surfaces and line the insides of hollow organs and glands. Epithelial membranes are classified by their number of layers. We have simple epithelium with one layer, and it's specialized for transport of substances. We have stratified epithelium composed of multiple layers, and these multiple layers provide protection. So we have transport and protection offered by epithelial tissue, as well as numerous other things that we'll discuss. So epithelial tissues are classified by the shape of their cells. If they're squamous, they are flattened cells. If they're cuboidal, they're as tall as they are long. If they're columnar, they are tall cells. When you put the cell shape together with the number of layers, this is how you name the epithelial tissue. A good example will be like simple squamous epithelium. Some modifications can occur, such as in the columnar tissues that have goblet cells. Goblet cells secrete mucus and cilia that move in a coordinated fashion. This is important in like the respiratory system. So the mucus is moving out of the lungs and you swallow it is the digestive tract. Table 1.3 is again in blue in your book and peach here. It's found on page 12. And it is the summary of the epithelial membrane. So you can refer to that for your reference. And we see that there's simple epithelia and stratified epithelia. Simple being one layer of either flat cells in the case of squamous, cube-shaped cells in the case of simple cuboidal. We also have simple columnar. Then we have simple ciliated columnar, and also one called pseudostratified ciliated. This appears to be stratified, but in fact is only one layer. Hence pseudostratified, falsely stratified. Then you have the stratified epithelia, one or more layers, and the topmost layers are the ones that have that shape to them, like stratified squamous epithelium. You've got the keratinized and non-keratinized versions based on where they're found in the body. You have stratified cuboidal epithelium and then transitional epithelium. So refer to that table when looking at the structure and function and location of these tissues. Here's some example of some of the structure and functions of epithelial tissues, such as simple squamous epithelium. This allows for rapid diffusion as the alveoli of the lungs. Simple cuboidal epithelium allows for the secretion of substances, as in various glands. And the simple columnar epithelium allows for the absorption as found in the wall of the small intestine. And those are pictured here in figure 1.12, which is a nice big illustration in your book. You can see the simple squamous is A at the top, the simple cuboidal is B, and the columnar is going to be C. For the stratified examples, we see that there are going to be tissues held together by structures called intercellular junctions, and in your book it is showing a stratified squamous non-keratinized epithelium. And so as you look at that illustration, which will probably be featured in the next slide, in order to provide that protection, these cells need to be held together tightly. And they are thus too close together to house blood vessels. And so the epithelium are nourished by connective tissues beneath. However, as we get farther up towards the surface layer, these cells become farther away from the connective tissues and they die. And it's okay because these top layers are for protection and they get sloughed off anyway, and then they're replaced by the basal layers below. So epithelial tissues are attached to connective tissues by a basement membrane. And so let's go ahead and take a look at that picture of the non-keratinized stratified epithelium. And that basement membrane is going to be that junction between the pink tissue in figure 1.13a and then the stained blue tissue beneath it. So that basement membrane is the union of the epithelium and the connective tissue below. And so these lower cells are going to be nourished by the underlying connective tissue, whereas these cells in the upper layers are far enough away that they are dead. So the non-keratinized membranes that we just saw have living cells in all layers, after I just told you that they're all dead. But anyway, they still get sloughed off, and I guess they're not nearly as metabolically active, so my apologies for that. And then the keratinized ones, they are truly, truly dead when they are up at the top because they're filled with a water-resistant protein called keratin. And so they easily slough off. I guess that's why, maybe it hurts a little more when you tear the epithelium of your mucous membranes of your mouth and your esophagus than when you maybe scratch your skin, etc. But I'll learn something today. So all these cells in the non-keratinized are alive. How about that? Squamous surface cells. That's so cool. But then see how beneath that is the mitotically active germinal area. So this is where the cells are going to be dividing, and they'll replace these surface cells as they get sloughed off. And here is your keratinized layer. And so truly up here at the top, that keratinized layer, they are dead, dead, dead. And then at the bottom here, they are alive and metabolically active, and they are going to be connecting to the underlying connective tissues of that dermis. All right, so epithelial tissue can also form glands. And so the two major types of glands are exocrine and endocrine glands. And so we'll look at the exocrine gland first. They are derived from epithelial tissues, and the secretions are transported by ducts, which are passageways, kind of like your air conditioning unit that carries AC from your air conditioning unit all the way through your house. And so the things that the exocrine glands are carrying through their ducts are like the lacrimal secretions, which are your tears, sweat secretions, sebaceous glands, which are oil, digestive enzymes, and the prostate, which helps to finish forming the sperm into semen. The secretory, or the part that secretes the exocrine substance, may be tubes or acini groups. So if they're acini groups, they're going to be more bulbous shaped than the tube. So sweat glands are a good example. Eccrine and merocrine are more numerous than the apocrine. Eccrine or merocrine secrete what we call a salty sweat, and they are involved in thermoregulation and are found basically all over your skin, except like on your lips and nipples and a couple other spots. Apocrine are located in the axilla or armpits and your pubic region, and they produce a protein-rich sweat that bacteria feed on, and that's not the only reason for those sweat secretions. The apocrine secretions are for like pheromones, and so they start to turn on during puberty, which can tell you a lot about the way you start feeling at puberty and the way you start smelling, because as the apocrine secretions are eaten by bacteria, they start to smell. Okay, so here is some pictures for figure 1.16 that shows the structure of exocrine glands. Exocrine glands may be simple invaginations of epithelial membranes, or they may be more complex derivatives. So in the first two, they're pretty simple. There's only one duct. And then as we get to the acinar, it's getting a little more complex. This is called a simple branched acinar, meaning that each of the branches only has one tubes. But then sometimes you're going to get even more complexity than that, like with mammary glands, etc. All right, now let's look at the endocrine glands. These are also derived from epithelial tissues, but they lack ducts, and therefore they secrete their products into the capillaries within the body, and then the hormones will be transported throughout the body, where they will then interact with the cells, and the cells that have receptors for them will be stimulated to perform the directions that the endocrine hormone controls for. And so the structure of endocrine glands will be described in Chapter 11. And examples include many hormone-producing glands, such as your thyroid and your adrenals. Let's see, we've got your pineal gland, your pituitary gland, etc. So this is actually figure 1.15, and this is the formation of exocrine and endocrine glands from the epithelial membranes. Note that the exocrine glands retain their ducts that can carry their secretion to the surface of the epithelial membrane, whereas endocrine glands are ductless and they secrete their hormones into the bloodstream. So they all arise from that epithelial layer. All right, the fourth primary tissue type is called connective tissue. And this is characterized by a matrix made up of protein fibers, extracellular material, and specialized cells. There are four major categories of connective tissues: the connective tissue proper, cartilage, bone, and blood. Connective tissue proper is composed of protein fibers and a gel-like ground substance. There are several subtypes of connective tissue proper. Loose and dense are examples. So in the loose connective tissue proper, the collagen fibers are scattered loosely with room for blood vessels and nerves. For example, the dermis of the skin. In dense regular connective tissue, densely packed collagen fibers with little room for ground substance occurs, and this is found in tendons and ligaments. I think of the dense regular as kind of like a rubber band. You can flex and extend it, but you can't carry it too far or it'll snap. And the difference here is that with the lack of blood vessels for the dense connective tissue propers, they're not going to regenerate as easily or at all. So if a ligament or a tendon is torn, then it stays torn until it's like surgically repaired. Whereas the dermis can, for the big part, repair itself somewhat. It still will scar with collagen fibers laid down, but it can form that scar and heal itself, whereas the dense regular connective tissue cannot. So here we see the dense irregular connective tissue listed out in terminology here in the literature, and we can see that it's got densely packed collagen fibers in various arrangements to resist forces, and there will be room for blood vessels to move through it. And then in this illustration here, figure 1.17, this is a loose connective tissue called areolar connective tissue, and this is what's found in the upper layer of the dermis. This is the layer that is going to nourish the epithelial cells above. So this can largely regenerate as long as with the epithelium, as I told you. The dense irregular is going to scar, and the dense regular is going to basically remain injured because of that lack of blood flow. And then we also have another type of connective tissue proper called adipose. Adipose tissue will store fat in the cells called adipocytes. And in figure 1.19, it describes adipocyte tissue containing the large central globule of fat surrounded by the cytoplasm of the adipocyte. And so nutrients are stored in adipose tissue that are high in energy, but also like heavy metals and PCBs and things like that can be stored in the adipose tissue as waste products that the body doesn't know what to do with. All right, so the next type of connective tissue is cartilage connective tissue. And so this is composed of cells called chondrocytes surrounded by a semi-solid ground substance. And cartilage serves as the template for the skeleton during bone development, and it's found in joints to provide a gliding surface for bones. So your slide here has connective tissue proper down for, the cartilage there, but generally what we consider cartilage is supporting connective tissue. And the other type of supporting connective tissue that we'll see is the bones themselves. And so up here, if you ever see supporting connective tissue for cartilage and bones, don't be surprised by that. That is a very common grouping for them. So with the bone, we have cells called osteoblasts that trap mineral salts, forming concentric layers of calcified material around a canal filled with blood vessels and nerves. Once the matrix is hardened, the cells are called osteocytes, and they live in spaces called lacuna. The dentin of a tooth is similar to bone and is made by cells in the pulp, and the outer enamel is harder than the bone or the dentin. So in figure 1.20, it shows the structure of the bone. In 1.20a, shows a diagram of the long bone. 120b shows the photomicrograph of the osteons, and figure C is going to show the drawing of that photomicrograph. And you can see the central canal with the artery in red, a vein in blue, and a nerve in yellow. And then these sheets of bone called lamella are going to be built by the osteoblasts. And then once the matrix surrounds the osteoblasts and hardens, the osteoblasts mature into osteocytes, and they reside in their lacuna for as long as they live. That's their retirement. And so here's a tooth that, I don't know why, but that picture makes my teeth hurt, seeing it shaved in cross-section like that. Ouch. We know that teeth are highly innervated, right, by that big old trigeminal nerve. And so let's do our best to take a look at the cross-section without hurting too bad. And so the pulp here is in the center, and the root of the tooth is anchored by cementum, which is a calcified connective tissue that helps anchor the tooth in its bony socket. And then we have the dentin that is going to be surrounded by the hardened enamel. So as you lose the enamel, if you have like soft teeth, that is what causes the soft teeth, and then it can break. And one last thing before I go is I have learned from teaching like zoology that the teeth are actually made of like dermal bone, which is why they're a bit different than your skeletal bone. It's like a different embryonic origin, actually from your skin, like your dermis of your skin. And so that's pretty neat. Like deer antlers, you know how they're bone? Well, they're like skin derived rather than skull or skeleton derived. Which is probably why they can shed them so easily and regrow them every year, and they're covered in that velveteen as they do regrow them. So there's a fun fact. All right, so next up we have chapter 1, section 4, which is going to cover the organs and their systems. So an organ is composed of two or more tissues that serve different functions in the organ. And the example they give you of an organ system to just kind of illustrate the importance of tissues is going to be the skin, the integumentary system. And this is good because the skin is the largest organ in the body, and it's the one that you see when you look at somebody. And so it has all 4 primary tissues as well. So check off all the boxes, a really good example organ system. So the 3 layers of the integumentary system are the epidermis, dermis, and hypodermis. And the epidermis and dermis are what are considered like the integument, and the hypodermis is below the dermis, and we consider that the subcutaneous layer. So you might hear the term cutaneous membrane in your studies or have already heard it. And so that is going to refer to that integument, as I mentioned, the epidermis and dermis, and then the subcutaneous layers, the hypodermis beneath. So let's take a look at the tissues that are in the epidermis. This is going to be keratinized squamous epithelium. And so it is going to protect against water loss and abrasion. And recall that this is keratinized, so all the cells at the top are dead, dead, dead. and they slough off, and of course they're replaced by the cells that are undergoing mitosis in the deeper layers. All right, deep to the epidermis is the dermis. Notice that the epidermis sits above the dermis. That's that prefix meaning there. And the dermis is composed of two layers, actually, of connective tissue. This slide only includes the dense irregular, but when we go back to that areolar connective tissue earlier in figure 1.17, that's part of that loose connective tissue I was talking about. That's going to make the top papillary layer of the dermis. And then deep to that is going to be the dense irregular connective tissue, which makes up the majority of your dermis anyway. And it is going to contain the exocrine glands and hair follicles sense receptors that derive from the epidermis but go deep into the dermis. It also is going to contain blood vessels, and it's going to have a lot of collagen that is going to be resistant to tearing, and it's going to be very tough. It's also going to hold on to water, so extremely important tissue type there. And then the hypodermis, which is deep to the dermis, hypo below, we also call that the subcutaneous layer. It's going to contain adipose tissue. And so, as mentioned before, it's energy storage, and it's also padding, so like shock absorption, and it insulates against the loss of body heat. Figure 1.22 is the diagram of the skin, and it says here that the skin is the organ that contains all four types of primary tissues. And so, in this illustration, we can see all four of those types, whereas if you notice in this slide here, we see that we've got the epithelial tissues in terms of the keratinized squamous, and then we have the connective tissues in terms of dense irregular and adipose, but we haven't mentioned like muscle tissues or nerve tissues yet. So looking here at the hair follicle, you'll see that there's a smooth muscle that is going to be connected to that hair follicle and the epithelium. And so when this muscle contracts, it causes the hair follicle to stand up, and that's what gives you your goose pimple. So it's called the erector pili. Erect meaning to stand straight up, and pili stands for hair. That's the muscle tissue. You can see the epithelium up at the top with that epidermis, that stratified squamous. And then you can see the dermis has the papillary layer, which is squiggly looking. Papilla means nipple. And that's going to be areolar connective tissue, which is the connective tissue of the papillary layer, and then the dense irregular connective tissue will make up the rest of the dermis as part of the reticular layer. And then strewn all throughout the dermis, you can see nerve endings, and they even go up and touch the epidermis because the skin is a very important sense organ. And then deep to the dermis, you can see the hypodermis, which is going to have your adipocytes. And then all throughout that, we also see blood vessels. So that is a great little slice of organ there, showing all four tissue types and subtypes of the different primary tissues. Okay, so now we'll talk real quick about stem cells. Stem cells are going to be involved with tissue development. And tissues are composed of highly specialized cells that arise from three embryonic germ layers: the endoderm, the mesoderm, and the ectoderm. Endo means within, so it's that innermost layer. Meso means middle, and then ecto means outer. And so the endoderm is going to form like the lining of your digestive system, your respiratory system, and a lot of like internal glands. So that's why it's endoderm, and that's going to be largely epithelial tissue. Your mesoderm in the center then is going to fill in like that body cavity with like connective tissue, so like bone and cartilage and blood. It's also going to give rise to organs. And then the ectoderm is going to be the outer layer that gives rise to the epithelium of your skin. It also folds inwards in the neural tube and forms the central nervous system. All right, so there are several types of stem cells, and they include zygotes and adults. So zygotes are going to be the beginning of your embryonic development, the first few hours of your embryonic development. So as soon as the egg is fertilized by the sperm, the zygote forms. And so the cells in the zygotes are totipotent, and this means their cells can become any type of cell. They can differentiate into anything. And these are the true stem cells. Now, how do they become what they're going to be? And that has a lot to do with if those cells are located in the endoderm, the mesoderm, or the ectoderm. I always tell my students it's location, location, location. So wherever they're located in the embryonic germ layers is going to determine whether they're going to be the lining of, your digestive tract or your respiratory tract, or whether they're going to make up connective tissue, such as in the mesoderm, or if it's going to become the epithelia of the skin or the nervous system if it's in the ectoderm. So these are true stem cells. They have the ability to become whatever. And it's location, location, location. But once the cells begin to differentiate, which means they become the cells that they're specialized to be based on their location, a few adult stem cells are retained to allow for cell replacement, because we have to still be able to regenerate our tissues. So adult stem cells are limited to a narrow range of possibilities, but they can become several related cells and are thus called multipotent. So bone marrow cells, for instance, can become any type of blood cell. Embryonic stem cells are pluripotent, and they can form any type of unrelated cells. Figure 1.23 shows the stem cells in the bulge of a hair follicle. And so the stem cells in this region migrate to form the differentiated cells of the hair follicle, the sebaceous gland, and the epidermis. All right, looks like last but not least, we look at all the organ systems briefly in a table. It's going to be table 1.4. And so humans are going to have 11 interrelated organ systems. And here you can see that those are going to be the integumentary, which was our example organ system for this chapter. And then, of course, the nervous system, endocrine system, skeletal system, muscular system, circulatory, also known as the cardiovascular system, the immune/lymphatic system, respiratory system, urinary system, alimentary or digestive system, and the reproductive systems. And so you can take a look at the table here in the PowerPoint and also in your book to see their major organs and their primary functions. All right, so we are watery creatures. We're 65 or more % water, and so the last couple of paragraphs in this chapter talk about the body fluid compartments. And so our water is going to be held intracellularly and extracellularly. If it's intracellular, it's going to be the area inside the cells, and it will contain 65% of total body water. So most of your water should be in your cells. The extracellular is going to be the area outside of the cells and will include your blood plasma in your blood vessels, as well as your interstitial fluid. Interstitial fluid is also called tissue fluid, and that's what surrounds all of the cells outside of the blood vessels. Both body fluid compartments are filled primarily with water and are separated by membranes, and there is selective movement of molecules and ions between the compartments through the cell membrane. And so we'll start talking about the chemical composition of the body in Chapter 2, and I believe Chapter 3 starts our cells. And so you can stay tuned. This is kind of building us up for moving forward in the book. So thank you for listening, and have a blessed evening or morning or whatever time it is for you. Figure out how to stop this thing. There we go. Bye.